a little girl. My name is Gregor Mendel. Today is a good day to go back in the time and remember all the good things I did when I was younger. I'm going to lie you with all the awesome things I discovered when I was bored with my plants. For thousands of years, farmers have been selectively breeding their plants and animals to produce the more useful hybrids. It was somewhat unheard of mixed process since the actual mechanism governing inheritance were unknown. Back in the great 1890s, the invention of better microscope allowed us, the biologists, to discover the basic facts of cell division and sexual reproduction. To focus of genetics, research then shifted to understand what really happens in the transmission of heredity and traits from parents to children. A number of hypotheses were suggested to explain heredity, but I was the only one who got it more or less right. My awesome ideas were published in 1866, but weren't recognized until the 19th, which was long after I passed away. When I was kinda young, I spent my entire time doing basic genetics research and teaching in high school for students like you. Some were topics of mathematics, physics, and also Greek. But when I was getting a little bit too old, I became the abbot of a monastery and put aside all my scientific work. So, through the selective crossbreeding of two common pea plants over many generations, I discovered that certain traits show up in offspring without any blending or parent characteristics. So I observed 7 traits that are easily recognized and apparently only occur in one of 2 forms. The number 1 was the little cute plant I have was either purple or white. And the number 2 was the position it has was axial or terminal. My plant was short or long. The 4 was that the seed has a shape that is round or wrinkled. And also my seeds were either yellow or green. And the number 6 was the pot shape was inflate or constrict, and 7, my pot was either green or yellow. These traits do not show up in offspring plants with intermediate forms, and that was critically important because the leading theory at that time in biology was that hereditary traits blend from generation to generation. So most of the leading scientists in the 19th century that weren't as cool as me accept the blending theory. Also, my friend Darwin proposed another equally wrong, known as pangenesis. This held that hereditary particles in our body are affected by the things we do during our timeline. These modified particles were through to migrate via blood and to the reproductive cells and subsequently could be inherited by the next generation. So I pick up common garden green plants for the focus of my research because they can be grown easily in large numbers and their reproduction can be manipulated. Pea plants have both female and male reproductive organs, so as a result, they can be either self-pollinate themselves or cross-pollinate with another plant. In my experiments, I was able to selectively cross-pollinate plants with particular traits and observe the outcome of over many generations. This was the basis of my conclusions about the nature of genetic inheritance. So I conclude 3 things. The first one was that the inheritance of each trait is determined by genes that are passed on to the descendants unchanged. The second one is that individual inherits one such unit from each parent for each trait. And the third one was that a trait may not show up in an individual but can still be passed through the next generation. So my observations can be summarized in two laws or principles. The first law is that according to my principle of segregation for any particular trait, the pairs of alleles of each parent separate and only one allele passes from each parent onto an offspring. And which allele in a parent's pair of alleles is inherited and is a matter of chance. We know that this segregation of alleles occurs during the process of sex cell information. And my second principle was that according to my other principle, the one from independent assortment, different pairs of alleles are passed to offspring independently of each other. The result is that new combination of genes present in neither pattern as possible. For society, manipulating the genes of a plant is correct because it's for science research. But if I had manipulated an embryo, everyone would have complained and hated me. In conclusion, one of the reasons that I carry out my experiments with pea plants was that I could observe inheriting parents in up to 2 generations a year. Geneticists today usually carry out their breeding experiments with species that reproduce much more rapidly, so that the amount of time and the money that is required is significantly reduced. Fruit flies and bacteria are commonly used as proposed now.